Hello and welcome everyone to this video podcast for evolution and diversity. In this video podcast, we're going to focus on the methods of speciation. So if you remember in the last class, in the last set of videos that you looked at, we focused on the concept of species and the three different ways that species could be defined: morphospecies, biological species, and phylogenetic. And we also talked about how populations could become reproductively isolated. So today, instead of focusing on what a species is, we're going to focus on the pathways that lead to new species. And so we want to talk about the two main pathways, allopatric speciation and sympatric speciation. And then we want to spend a little bit of time talking about what happens when you have two isolated populations come in contact with each other. Now, before we start talking specifically about allopatric and sympatric speciation, let's begin with just a simple definition of each of those two types of speciation. So we have allopatric speciation and we have sympatric speciation. In general, we can think of allopatric speciation as any event that leads to new species based upon a geographic separation. You have one population and they become geographically separated from each other, and now each side can evolve into new species. So sympatric speciation is a little different. It's when speciation occurs within the same geographic area. There is no separation between the two different populations. They're intermixed within the same area, but still two different species can be formed. Okay, so we're going to now focus on allopatric speciation, but before we do that, I'd like to define a term, and that term is biogeography. And biogeography is the study of how species and populations are distributed geographically. This field of science is particularly interested in studying allopatric speciation. Now, there are two types of allopatric speciation pathways, and the first is called dispersal, and the second is called vicariance. I want to spend a little bit of time talking about each of these pathways. All right, so let's begin this discussion by focusing on dispersal. So with dispersal, there are three steps, and really these are going to be the same three steps that we see with vicariance. But the first is there has to be some sort of geographical isolation. Second, which is related but it's quite different, is that there has to be a genetic isolation. Another way of saying that is that there can be no gene flow. And then once you have these two separate populations that are geographically isolated and genetically isolated, then there must be some sort of genetic divergence. So with dispersal, what's happening here is you may have a population here, and then part of this population leaves the main population and begins a brand new population. So this would be population 1. And this would be population 2. So this would be a geographical isolation. They're in different areas. And this will sound familiar to you because this is what we talked about previously when we talked about genetic drift. What we're talking about is the founder effect here. The founder effect creates this geographic isolation. Then the next step, as I said, is genetic isolation. The populations have to be far enough apart from each other where there is not a regular gene flow between the two populations. In fact, with this kind of speciation, the further apart these two populations are, the greater the chance that speciation will occur. And that is true for two reasons. The first is it will reduce gene flow. The further they're apart, the less likely they're going to come together and share alleles. And the second reason is a different environment. Two different environments that are 10 miles apart from each other are probably not going to be that different. So it's going to be less likely that natural selection can be a factor. However, if you talk about a 500, 1,000, 2,000 mile separation, then you're talking about quite a bit of a difference in the environment. Maybe it'll be colder, maybe it'll be warmer, maybe it'll be wetter, maybe it'll be drier. Lots of different things can happen the further apart they are. And again, I said that's because it allows for natural selection. to occur, or increases the chances of natural selection. So this is important, the distance between these two geographical locations. And that brings us to this next idea of genetic isolation. The further they're apart, the less likely they are going to be able to have gene flow between those two. In order for dispersal to work and actually lead to a new species, the gene flow has to be eliminated between these two populations, or at least greatly reduced. Now, genetic divergence has to set in, and what I'm talking about here is things like natural selection, genetic drift, gene flow, and mutations. We already talked about gene flow, and that's going to speed up this genetic divergence if there's not genetic flow between these two populations. Genetic drift in this example has already been taken care of because it's separated out these two populations with the founder effect. But natural selection can also speed up genetic divergence between these two populations. And mutations that occur in either of these populations will also speed up genetic divergence and ultimately speed up the process of speciation. I'd like to spend a little time looking at a specific example of ground finches found on an island called Daphne Major. Just to give you a point of reference here, Daphne Major is a tiny island. It's really, compared to these other larger islands you see here, it's just almost a little speck. So I kind of put a little picture in here to give you an idea what the island might look like. But it's a small little island, but it's a very protected island for these finches because there are really no predators there. So it makes for a nice island for these finches to populate during some months of the year. So let's draw Daphne Major here. It's a circular island, I guess, so it's pretty easy to draw even, with my amazing skills. So this is Daphne Major, and I'm just going to put DM for Daphne Major here. These ground finches have a home island, and I'm just going to draw it here, and I'm just going to call it the home island. This home island is where the ground finches breed, so this is their breeding island. But for a few months of the year, these ground finches would migrate over to Daphne Major, and they would spend a few months here in between breeding seasons, but then they would always fly back to their home island to breed. Two scientists, Peter and Rosemary Grant, they studied the finches on Daphne Major, and what they discovered was that one year, 13 ground finches stayed on Daphne Major. The rest of the population went back to their home island to breed. The ones that stayed on Daphne Major, they stayed there and they breeded. So let's put that here. Stayed on Daphne Major and breeded with each other. And this is what dispersal is all about. Having a large population and then some of that population breaking away from this main population and forming a new colony. So this became the new colony of finches. And this over here was the original source. Okay, I went ahead and erased what we had here and redrew part of it up here because I want to be able to use this as part of the next discussion here. Because I want to go back and remind ourselves of what the 3 steps are for allopatric speciation using the dispersal method. So remember that first step was to have geographic isolation. So we've achieved geographic isolation because you started off with a large colony of ground finches that were part of this home base here, the home island. And then when they migrated to Daphne Major, remember a few of them stayed there. And then the rest came back over to the main home island to breed. And these ones that remained here started breeding with each other. So now you have a geographic isolation where you have 2 different populations breeding on 2 different islands. So the first step has been achieved. And this next step of genetic isolation should be considered next. So the fact that they're on separate islands helps keep them genetically isolated. It helps reduce gene flow. But another thing that's happening here, if we go back to a term we learned in the last class, this is a reproductive isolating method. 2 different populations in different habitats that breed in these different habitats. So reproduction between these two populations has been isolated, which helps isolate the gene flow between these two populations. So we've considered this step. Now this last step is also something we should look at, and remember this was genetic divergence. Has something happened between these two populations to have allowed one of them to be genetically distinct from the other population? Remember, this could be genetic drift, it could be gene flow, it could be new mutations have arose, or it could be natural selection. Now, two big things certainly happen in this population, and the first is genetic drift. And with the dispersal method, this is almost always going to be the case, where you form a smaller population from a larger population. So that should almost always create a little genetic differences between these two populations. But the Grants also discovered something else, and that is that there was some natural selection happening as well. And what they discovered is that the beak sizes in these two populations of ground finches were different. The finches on Daphne major, they had larger beaks compared to the beak sizes of the ground finches in the original colony, which had smaller beaks. And this corresponds to the available food sources in these two areas. Larger beaks on Daphne major provides an adaptive advantage to survival, to being more fit than having a smaller beak. And likewise, on this home island, having smaller beaks provides an adaptive advantage. So natural selection has occurred between these two populations. So this is one example of dispersal allopatric speciation. Okay, now I want to talk about the other pathway for allopatric speciation. And this, remember, is called Vicariance. Now with Vicariance, we also have those same three steps. We have to show geographic isolation. Then we have to show genetic isolation. And then finally, genetic divergence has to occur. Now the main difference between Vicariance and dispersal is that with Vicariance, the way we achieve geographic isolation is different. So let's draw a population here of this species. And remember, with dispersal, there was some sort of founder effect where a part of this population, a small part of this population, settled and formed a new population, a new colony, like we saw with the ground finches. But with Vicariance, something else is going to happen. And that is, there's going to be some chance event that's going to separate these two populations. So maybe a mountain forms between them, maybe a river forms between them, but some chance event, so why don't we just write that here, chance event, separates the population. Now, since these two populations on either side of this geographic event that happened, maybe, like I said, a mountain formed, these two populations now are not going to be able to easily breed with each other. And as a result, they will become genetically isolated from each other. They're in different habitats now. Again, it's a reproductive isolating mechanism. Now, over time, genetic divergences in these two populations can take place. So again, we could have genetic drift, where part of one of these populations is lost, and now you have a smaller population left, which would be an example of the bottleneck effect. It could be that one of these populations develops new mutations. And since there's no gene flow between them, you can't share those mutations on either side. So the next one corresponds with what I just said, and that is we could have gene flow blocked between these two now separate populations. And lastly, natural selection can affect the genetic diversity of these two populations to the point where they can now become distinct populations. So let's consider an example of Vicariance.
All right, for the example of Vicariance, I want to use an example that your book describes. I think it's a really good example, and so I'm going to just borrow the figures from the book to talk about this one. And for this example, we're going to focus on the diversity that we find in the Amazon rainforest. The Amazon rainforest is well known for its very rich diversity, but it's also well known for many speciation events that have happened over the past millions of years. And this bird here, called the trumpeter, is a good example of how speciation has occurred in the Amazon rainforest. These trumpeter birds, they can swim short distances and they can fly short distances, but for the most part, they spend their lives on the ground. Researchers who were interested in the speciation of the trumpeters collected DNA samples from these trumpeters all across the Amazon rainforest. They were able to generate a phylogenetic tree, which I'll show you in a moment, and they were able to estimate how long ago certain events occurred, certain speciation events occurred. And they did this by looking at the formation of the Amazon River and the Andes Mountains. So here's a drawing of South America 3 million years ago. And you'll notice that there is not an Amazon River shown in this picture. We do see this rather large lake and wetlands area, and we have fossil record of where this ancestral population of trumpeters were. Sometime between 2 million and 2.7 million years ago, the Andes Mountains started to form. And when these mountains formed, that helped create the Amazon River. And this Amazon River, as you can see in this picture here, ran right through the middle of this population of trumpeter birds. So this caused that first geographical event that isolated these two populations. And likewise, because there was little chance of gene flow between these two populations, because remember, they didn't fly far distances and they didn't swim far distances, that kind of kept them isolated genetically. Gene flow was reduced dramatically. And then what happened is, with further land shifts, partly by the Andes Mountains, but also because the Amazon River started to form these large tributaries. And in doing so, it split the population again, and you ended up with a third population of trumpeters that were geographically isolated from the other two populations. And that occurred between 1 and 2 million years ago. Then less than 1 million years ago, between 0.3 and 0.8 million years ago, we see evidence of more tributaries forming. Again, splitting these once three populations of trumpeters into now approximately 8 different populations of trumpeters. Again, each of these cases involves a population being geographically isolated from the other populations and becoming genetically isolated between those populations, because gene flow cannot occur between these isolated populations. So the first two parts of allopatric speciation using Vicariance have been achieved with this, right? We have the geographic isolation, we have the genetic isolation. Now, is there anything that can be done to increase the genetic divergence between these populations? Is there evidence of mutations? Is there evidence of natural selection, of genetic drift, or a block in gene flow? And so when they examine the DNA sequences, they can determine that indeed these did form different species. And so one of these events must have occurred to make these genetically distinct populations. Certainly gene flow was blocked, and because this is such a large area, one can only imagine that, yes, there are morphological differences and there are adaptive advantages that one could imagine being, say, near the Andes Mountains here versus being near the Pacific Ocean along these regions here. So certainly there has been some natural selection that has gone on as well. So let's look at the phylogenetic tree that was generated by looking at these different regions. And they were able to show different monophyletic groups and different species. Again, there was barriers to gene flow that prevented the different populations from interacting with each other and from breeding with each other. Various mutations, natural selection, genetic drift happened in these isolated populations that allowed these different populations to diverge from each other and ultimately become these different species. So to summarize allopatric speciation, it's important to remember that whether we're talking about dispersal or vicariance, that the two populations must become geographically isolated from each other. These two different populations now become genetically isolated from each other because there is a lack of gene flow between those two populations. Then, on top of that, if we see genetic drift or natural selection or mutations, that will help increase the genetic divergences between these different populations, either the dispersal or the vicariance method. All right, let's go ahead and move into the next category of speciation, and that is sympatric speciation.
Okay, so let's go ahead and start talking about sympatric speciation. And remember, the main difference with sympatric speciation compared to allopatric speciation is that with sympatric speciation occurs in the same geographic area. So similarly with allopatric speciation, we need to follow these 3 steps for sympatric speciation. But they're not going to be exactly the same as allopatric speciation. The first is that there is no geographic isolation. And this is a big difference, right? Because remember, with allopatric speciation, there was a geographic isolation. So you may have a population here with different individuals of the same species. So at least initially, the mating between individuals in this population is random. However, within this population, we still are going to see genetic isolation. Similarly, as we saw with allopatric, except this genetic isolation is a little harder to understand because there's not a geographical barrier preventing genetic mixing or gene flow. But over time, what happens in this population is that the mating becomes less random. So what I mean by this with less random mating is now, for instance, maybe pink individuals are more likely to mate with each other than pink with blue individuals. And same thing with blue and blue individuals. So it's not random anymore. And this is a way to get genetic isolation, right? You're still within the same population, but if similar types of species only mate with similar types of the same species, then you are isolating that genetic material within those similar types. And lastly, we will also see genetic divergence, just like we saw with allopatric. This population with these different types of individuals that are now mating less randomly may experience some sort of evolutionary pressure, natural selection, a mutation, genetic drift, or something like this. There are a few ways we can think about sympatric speciation working, but we're going to talk about 2 different ways. The first is disruptive selection, and the second is polyploidization. We're going to first begin by talking about disruptive selection. But before I do that, I'd like to define 2 terms that may come in handy as we're talking about disruptive selection. And these 2 terms are niche and ecotype. A niche is a range of ecological resources a species uses and a range of conditions that species can tolerate. And an ecotype is a distinct form of an animal or plant occupying a specific niche. Okay, so as we talk about disruptive selection as a way to get to sympatric speciation, we're going to use these terms, so keep them in mind. Remember that disruptive selection went from a range of traits, like so, to a selection of the extreme traits. And when we described this in the last unit of material, we said that disruptive selection often leads to speciation. So now we're going to come back to that and talk about how it does lead to speciation and how this kind of speciation is most likely of the sympatric variation. And as an example of disruptive selection, I want to talk about killer whales. Researchers have studied killer whales for decades, if not centuries. And we believe that killer whales were a single species. Granted, they have a very large distribution and they're found in all the oceans of the world. But still, we believe they were a single species. Researchers began to realize that you could identify different populations of killer whales based upon different sizes of the dorsal fin, different markings on the fin or on the the killer whale, the different sizes of the pods they swam in. And one researcher recognized two distinct populations. One he labeled transient, which is this picture on the top, and the other he called resident. The first thing that was noticed is that they travel in different group sizes. Transient killer whales traveled in group sizes of 2 to 6 individuals, whereas the resident killer whales traveled in groups of 10 to 25 individuals. He also noticed that there was a difference in size overall. The transient killer whales were larger, and the resident killer whales were smaller. This particular researcher also noticed that there was a very distinct feature on the dorsal fin, and that's on the back of the killer whale. And he noticed that the transient killer whale had a pointy dorsal fin. The resident had a dorsal fin that was less pointy. Not very scientific sounding, but less pointy. And it kind of had this like little curve on it. Now I didn't use the term, but I should. We would consider these two groups of killer whales to be ecotypes. So this would be best called the transient ecotype. And down here would be the resident ecotype. While both of these killer whale ecotypes lived in the same area of the Pacific Ocean, they did not intermix with each other. They did not interact with each other, and they did not breed with each other. So this was a very curious observation. There was no reason to expect that these two ecotypes of killer whales that lived so close to each other wouldn't be able to interact and breed with each other, because there is no geographical barrier between these two ecotypes. As time went on, more researchers were getting involved, and they noticed some other observations. And so I'm going to erase these so I can have a little bit more room. When one of these types of killer whales died or were found dead on a beach, they would examine the stomach contents. And from that, they could tell that these two types of killer whales, these two ecotypes, had a different prey source. The transient ecotype primarily ate marine mammals, like seals, sea lions, and other marine mammals, whereas the resident ecotype primarily ate fish, and mainly salmon. Now, their preference for these different food sources may very well have been a learned behavior. In fact, there's good evidence that because these killer whales live in a fairly tight family unit, that they do learn these kinds of eating behaviors and preferences. Something else that they noticed, which is kind of linked to what they eat, was their communication. For a very long time, we've known that killer whales can be very chatty, for lack of a better way of saying it. They have a very complex set of vocal calls, and you've probably heard some of these in various recordings. And the resident ecotype of the killer whale, like I said, they're very chatty, and they use this elaborate set of calls as a form of communicating with other killer whales in their pod, in their group, about the source of salmon or other fish that they happen to be hunting. They can also use it as a source of echolocation to zero in on a large source of a fish for them. So it's very important that they're chatty. However, in the transient ecotype, when they're trying to hunt marine mammals and seals and sea lions, they need to be sneaky, right? They're less chatty. I would make a horrible transient ecotype killer whale, because to be successful, you have to be less chatty, and you have to be sneaky, right? Because they have to sneak up on the marine mammals, the seals or sea lions that they're hunting. And so they have to have stealth qualities. That's probably a better word to use than sneaky, in order for them to survive. So over time, what has happened was that within this transient ecotype or the resident ecotype, certain behaviors were probably selected for that allowed them to feed on a preferred food source. A less stealthy, more chatty killer whale of the transient ecotype would not do very well and would not pass on their genes as effectively, would be less fit. A less chatty, a less vocally skilled resident ecotype would not do as well because they would not be able to find the fish they need to find. One other difference between these two is that the transient ecotypes tend to travel a much larger distance than the resident ecotypes. As I said earlier, these two types do not interact in the wild. They do not breed with each other in the wild. Now, if they are captured and in captivity, they can breed with each other. So it's possible that they haven't completely formed these two new species, but they're in this process of forming these two new species. But in nature, they do not breed with each other and they do not have progeny. So let's go back and think about our disruptive selection graph here. So remember, we went from a graph that looks something like this to a graph that looks something like this. These two extremes represent these two ecotypes. Over here, maybe it's the resident ecotype, and they have the behaviors of eating fish, more vocal, here eating mammals, and being more stealthy. Two extremes. Killer whales that could, say, eat both fish and mammals, or have both vocal skills and are stealthy, might be represented by this peak that's now gone, right? This ability to do both of them does not serve as much of a selective advantage as being able to do one or the other. So over time, these two ecotypes have been selected, and the intermediate, with this intermediate range, has been lost. Another way to think about this is that this once large population of killer whales have adapted to survive in different niches. The resident type has a niche of eating fish, of using vocal cues, of living in a larger pod of whales. This other ecotype, the transient, occupies a different niche. It has a different diet, has different behaviors. And again, this is selected for the two extremes because there are two extreme niches that are being occupied by these two different ecotypes of killer whales. That's all I want to say about the disruptive selection pathway for sympatric speciation. Now I'd like to spend a little bit of time talking about polyploidization. All right, so let's talk a little bit about polyploidization, and I want to define a few terms for you. The first term is, what is a polyploid? This is an organism that has more than two sets of chromosomes. Remember back to maybe a high school biology class or some other biology class that you may have taken, that most plants and animals, they exist as a diploid. And a diploid has two copies of every chromosome. Polyploids have more than two copies of every chromosome. So you and I, we have two copies of chromosomes 1, two copies of chromosomes 2, chromosomes 3, and on down the line. Polyploids might have 3 copies of all of those chromosomes. Now, it should be noted that polyploids are very common in plants and some invertebrates. Every now and then, there will be a human polyploid, but they die in utero and are spontaneously aborted. But in plants, extremely common. There are 2 types of polyploids, and we're going to focus on one, but I want to define both of them for you. The first is autopolyploid, and the second is allopolyploid. An autopolyploid is a polyploid formed from the same species. And it's almost always the result of an error in meiosis or mitosis. Allopolyploid is similar, except it's a polyploid formed from different species. And we'll talk more about these when you take genetics, but I do want to say a little bit right now about how they can form new species. And in particular, I want to talk about autopolyploids. That is, the polyploid that forms from the same species. And I want to walk through just a generic example. Let's say we have a plant species that is a diploid. It has 2 copies of every chromosome, just like you might suspect. It's going to go through meiosis, and you don't have to go back and review what meiosis is. Just know that the pathway of meiosis creates gametes, eggs and sperm. And in that process, it takes a diploid cell and creates a haploid gamete that only has 1 copy of every chromosome. Now, when this haploid gamete fertilizes with another haploid gamete of the same species, they produce a diploid progeny. This is just like we would see in mammals and in humans and in other animals. We're talking about plants here, but it's this similar process. Now, sometimes what happens, and again, this happens a lot more in plants, while it's going through meiosis, there's an error. And for the purpose of this class, we don't have to worry about what kind of error would lead to this, but you just need to know that it's an error. If you're interested to know more about how the error occurs, you can let me know and I can explain it to you, but for now, let's just call it an error. And when it makes a gamete, it makes a 2N, a diploid gamete, instead of a haploid. In fact, just so we can remember what's going on, let's write under this gamete. These are gametes that are forming. Now, when this gamete comes in contact with another 2N gamete, and so this is going to result in a 4N progeny. 2 diploid gametes will give you a 4 chromosomal copy number progeny, which we would call a tetraploid. So as a result of this, we have 2 species. We'll call this one species A, and we'll call this one species B. Now remember, a feature of sympatric speciation is that they exist in the same geographic area. So what is to prevent species A from species B from mating with each other? What is keeping them genetically isolated from each other? Well, let's talk about that and see why a diploid organism and a tetraploid organism cannot mate with each other. And what I'm going to do is I'm just going to work in the opposite direction here. Species A, which is a diploid, it's going to make gametes that are 1N, haploid, right? Because it's going to go through meiosis and it's going to make this haploid gamete. This tetraploid, species B, will also go through meiosis, and at this point there's no error in meiosis. That first error was just a fluke, right? We wouldn't expect it to always happen. And so a 4N individual, when they go through meiosis, they're going to create gametes that are 2N. If these 2 species try to unite their gametes, a diploid gamete with a haploid gamete, what's going to happen is it's going to make a 3N progeny, a triploid. And without going into great details, triploids tend to be sterile. And when you take genetics, you'll learn a little bit more why a triploid tends to be sterile. But for our purposes right now, what you need to know is that a diploid individual is capable of reproducing. A 4N tetraploid species is capable of reproducing with itself or with another 4N individual. But when they try to mate together, they produce this triploid progeny, and a triploid progeny will be sterile. This is an example of reproductive isolation where 2 fertile species produce a viable offspring, but that viable offspring is sterile. Gene flow is now blocked between species A and species B. So they're going to live in the same area, the same geographic area, but they are genetically isolated from each other because the progeny they make is sterile. That's all I want to say about polyploids and autopolyploids, and really all I'm going to say about sympatric speciation. We just have one more topic I'd like to cover in this material, and we'll talk about that next. All right, so now I want to talk about our last topic for today, and that is what happens when isolated populations come in contact with each other. So we may have population A here and population B here, and for generations, maybe thousands of years, they've never come in contact with each other. But now what happens when, for whatever reason, they do come in contact with each other? Maybe there's been some shift in land formations that have brought them together. Maybe waterways have opened up that have allowed these 2 populations to now come in contact with each other. Maybe humans have brought them in contact with each other. There's lots of reasons that isolated populations could someday share the same area and come in contact with each other. Well, there are 5 different things that could happen. maybe more, but we're going to talk about 5 of them. The first is that when these two populations come together, we could have a fusion event. So fusion of the populations. And what I mean by that is that these two populations freely interbreed with each other. Another thing that could happen is that one of the populations could become extinct. If both populations come together and they're competing for the same niche, it's possible that one of them, in fact it's quite likely, that one of them will be better at competing in that niche and they will outcompete the other population that came in contact with it and it will become extinct. Another thing that could happen between these two populations is reinforcement of divergence. And so if these two populations come together, and if the hybrids, so they do mate with each other, but if the hybrids are less fit, then natural selection will favor traits that prevent interbreeding. So maybe it'll favor a trait that makes the gametes have a mechanical problem where they cannot fertilize each other, as an example. Now the fourth possible outcome is that there is a hybrid zone formation. And so this one I think is a little more difficult to understand, but if you have a population A and a population B that come in contact with each other, there may be this area where hybrids form. So you get an AB hybrid, but it only forms in this area. It doesn't behave like the fusion explanation does, where it becomes fully interbreeding. Rather, there's just one small area within this larger geographic area that allows for hybrids to form. And the last one is the formation of a new species. So if species A and species B, these two populations come together and they breed with each other and they form this new species that doesn't breed with either A or with B. Now we end up with a total, in this case here, 3 different species because the hybrid forms this new species. All right, that is all I want to say about this last objective here, and that's all we have to cover for today's material. We talked a lot today about how species are formed. We talked about how allopatric speciation occurs and how sympatric speciation occurs, and then we ended with talking about what happens when these distinct populations that have gone through speciation have come back together. All right, if you have any questions at all, please make sure you come see me or send me an email. If not, I will see you in class. Bye for now.